Hello.
Okay. All right. Thank you.
An unsettling computer voice. Hi, everybody. It's nice to be in my office talking to you. Let me just adjust this. So, to be totally transparent, I'm here to try to convince you to apply to UT Dallas. And I'm going to be honest. You probably not heard of us. And that's fair. We're a very young school. But Texas is this wonderful thing. So you may have lots of preconceived notions about Texas, but the one thing that is 100% true is there's just a ton of money and it's all underground. So you don't really have to be particularly smart to have money in Texas. But fortunately, we did have some pretty smart people who decided that, you know, in a state with 29 million people, probably need more than like 2 major public universities producing science. So they have very aggressively expanded the number of PhD granting institutions, both in quantity and quality. And about over the last 10 years, we've gotten about $2 billion worth of stuff, $2 billion worth of financing. And now we are doing very well for ourselves. And then, of course, I am personally from Indiana, which is not necessarily a whole lot better than Texas, but I understand, you know, I'm a Midwesterner. I grew up in the Midwest. I did my postdoc in Chicago and my PhD in Notre Dame, and I went to Indiana University. So I was very apprehensive to move to Texas myself. And I really, really love Dallas. I can't speak to the rest of the state. They do seem kind of nutty, but Dallas is wonderful. I mean, it has been an amazing city. The weather's beautiful. I don't know if you can see it out there in my office. Like I've got trees still with leaves on them. And it's really nice. It's like 70 degrees. I guess it's going to get cold tonight, but it's really nice. But let me tell you about the science we're doing. So I'm going to try to share my, we'll see if this works. Keynote share. All right. We're unable to connect to Zoom. What? That's weird. You're sharing a screen. I'm sharing it anyway, so that's bizarre. So let's just pop this up, press play. Okay. Do you see my slides or do you see something else?
We see you, the slides.
Great. Okay. I want to talk to you about a project that I thought was really neat. It's sort of like a little passion project that I had of making viruses hot, but literally hot. This was all done before COVID, so we were doing this sort of neat stuff. And then COVID, of course, changed our research direction into something quite different. We've now moved very aggressively into immunology. But I'm going to this is sort of a long story of how we've done this. And this will all make more sense, I promise, as it goes along. But basically, right quick, this is the University of Texas at Dallas when I arrived. It was pretty gross, not gonna lie. That there was not a lot going on. So I moved in somewhere down here in the middle. And then, you know, $2 billion later, we built a huge, massive structure. Now this is all built up. This is a rapid transit stop up here. It's a train that directly connects you to the airport and then to kind of a cute little town center up here. This is sort of like a dining area. You can see my mouse, right? I'm not just wiggling around. This is our research lab right here. It's a research facility. And yeah, it's been pretty great. And the university continues to expand. And right now, I'd say there's no application fee. You don't need to submit GRE scores. We're particularly interested really in just looking at the whole package, your grades and letters of recommendation sort of thing. And the trip out to Dallas is free, so you can find out if it's just a bunch of tumbleweeds and people with 6-shooters shooting, but which is not. Maybe the rest of the state is, but not Dallas. So definitely reach out to me if I've piqued your interest. Our research group focuses very broadly. One thing that we do tend to have in common are viral nanomaterials. And so we do have this sort of thing that I'm not going to get into on crystalline machines. But what we've gotten into, particularly with the pandemic, is developing new vaccine adjuvanting materials. So materials that sort of allow us to get away from booster shots and even get away from injections. So essentially making proteins, stabilizing them with metal organic frameworks that are so rigid and robust, and they protect the delicate protein structure, that you can actually shoot them out of a gun, which is really on brand for Texas. But I built it in my living room over the pandemic when we were all locked out. It's a pneumatic gun that can fire microparticles of vaccine. And the ZIF actually promotes an adjuvanting effect that improves B cells and stuff like that. And I won't talk about that or the supramolecular, macromolecular MRI contrast agent, which I think is really neat. But what I will talk about is this stuff in the lower left-hand corner, this thing we call photophage, all right? And essentially the idea is to control, originally was to control viral nanostructures using light. And that has since evolved into now in a cancer immunology project. And what we originally, the original idea was always been to design biomaterials, sort of take natural materials that you find in the world, and viruses being one of them. And there's a lot of great reasons to use them, and incorporate chemical components that respond to external stimuli like lights or magnetic fields, or internal stimuli like redox processes and enzymes and changes in pH, right? And one of the things that sort of drew me towards nanomaterials was that there are a number of reasons, but naively at the very beginning, I was really interested in this idea of nanomaterials being passively capable of accumulating in fast-growing tumors, thanks to something called the enhanced permeability and retention effect. And basically what the theory goes with that EPR effect is you've got a tumor, tumors grow fast. In order to accommodate that fast growth, the vasculature has to grow fast. And the epithelia that line the vasculature is not the best. It's not the best built. It's more like your typical Chicago roads. It's got holes all over the place. And the holes are the perfect size for your tires, amazingly. And just like the roads there, the epithelia are perfect size for nanoparticles. And so when they leave the epithelia, they kind of get stuck in between the cells, in this sort of like no man's land, sort of this extravasating into this space. And it's like falling through ice. It's hard to get back out once you go in. And so things that are above 20 nanometers, but below 300 nanometers tend to passively accumulate, right? So you want a nanoparticle, if you're going to do this, you want a nanoparticle that is at least 20 nanometers in size. Now, the problem is your kidneys can only excrete things that are less than 6 nanometers, right? So the kidney's a remarkable organ, but it's essentially a mechanical filter. And nanoparticles that are larger than 6 nanometers don't go through it. Instead, they accumulate in the liver. And solid metal nanoparticles like gold or silver or things you might classically associate with nanoparticles, they go in the liver and there's really no way for them to break down. Gold nanoparticles are really, really robust. Silver can break down, and colloidal silver may not be the best for you. At the end of the day, they may not cause any harm, but the FDA is very hesitant to approve anything that will go into your body but will never come back out, seemingly. So our interest sort of evolved in, like, we can do polymeric nanoparticles, which can biodegrade. But then we have, nature, who makes these remarkably persistent nanostructures. I mean, these things are, as we know now, kind of a pain. And COVID is actually fragile compared to a lot of viruses in nature. You have viruses, the tobacco mosaic virus, for instance, can live in the dirt for 100 years and remain infectious. And some of these are other plant viruses. And in fact, the viruses that infect plants and the viruses that infect other bacteria called phages are some of the most tough proteins. I mean, they can survive extraordinarily high temperature. They can survive chemical conditions. You can do actual chemistry on them in mixtures of DMSO and DMF, because they're so robust, and they have to be, all right? Like plants are outside exposed to the sun. They have no convenient way to get into a plant, right? Plants don't have, mouths or mupal membranes. So, they have to be really robust and be able to bide their time until they can get in. And the same thing goes with phages. And so bacteria get infected with viruses, and the viruses that infect bacteria are called phages. And then the same thing, they have to be really, really robust. And one phage in particular called Q beta that I kind of saw and I kind of fell in love with it because I'm a trained physical organic chemist. And, if you've had physical chemistry and organic chemistry, you combine the two. Yeah, right. So, but I loved it, right? And as a physical organic chemist, I've come to really appreciate these sort of neat structures and the copious amounts of what we see here. We've got thiols. So you see these disulfide bonds, and you see these other things in colors. These are primary amines. And so you can do a bunch of chemical reactions on these amines, and you can differentially do reactions on these disulfides. And you can see that there are two different types of disulfides, right? There's this disulfide that comes together and it connects 5. So there are 5 proteins that come together and it makes a pore, right? So it's a pore, it's like 2 nanometers. And then it's got another one, it's another pore. There's 6 disulfides, and that's 3 nanometers in diameter. And on the inside of that virus, that capsid, actually, there's RNA. And we all know RNA is very sensitive. Not for viruses. Viruses actually can make RNA that's very, very robust, and they protect it very well. But RNA also is able to hold on to drugs, right? So a lot of cancer drugs bind to DNA, and that's because cancer drugs have a greasy part, usually. They can bind to the greasy bases. And then they have like a cationic, like an amine that gets protonated, and that can interact with the backbone. And so because this Q beta contains a tangle of mRNA, you actually can kind of poorly, not perfectly, but sort of tightly hold on to drugs. It's sort of like a supramolecular container, right? And so this was what we found actually, is you could take a doxorubicin, which is a very popular cancer drug, and it'll go through the holes, it'll go through the pores of Q beta, and it'll get stuck in the RNA, and it'll get stuck there, right? And so this was Candace Benjamin. So Candace is actually a New Yorker, and I never held that against her, but she was always so nice and very, very, very brilliant. And she found this out, that you take doxorubicin and you mix it in with our virus, you actually get, now they go through the pores and then they get stuck. They get stuck in the virus and they don't leak out. So you can see, you just wait 1 hour, 2 hours, you just see if it comes back out, right? You take the virus, spin it down, pellet it, put it in a new solution, just see if the doxorubicin starts coming out. It's very obvious if it comes out, because doxorubicin, when it's in water, is fluorescent, it glows red. But when it's stuck in the DNA, it's quenched. It doesn't glow red. So we just look for fluorescence. And we found that actually, you can keep it in buffer for a really long time, and it doesn't come out until you heat it. So that heating, of course, gives it thermal energy. That interaction, that nice cozy interaction with the RNA is temperature sensitive. So once the temperature goes up, that binding becomes, then other forces start to take over and it comes out. When we thought, oh, that's great, we have a way to treat a patient. All we have to do is give this to them. And then once the patient is loaded up with a drug, we heat the patient up to 100 degrees Celsius and they're cured of the cancer, as well as, any other conditions they have. And so obviously that's not a really great therapeutic approach to boil your patient. So we had to come up with a different way to trigger the release using heat, but in a way that was very, very specific, highly localized, in a way that, you didn't have to heat the whole person. And so we settled in on this idea of using gold nanoparticles as sort of a photothermal antenna. So the light will hit them and they'll get hot. And so why gold nanoparticles? First, gold has this really wonderful interaction with sulfur. So if you've got gold and sulfur, they just love each other. If you ever have a wedding ring like I've got, and you go to a sulfur bath, like you go to like Yellowstone, or you go to Rotorua in New Zealand, or any of those other fun places, and you go to a really sulfurous thing, you get your gold ring in there, it'll change color. The sulfur really binds to gold really tightly. and we've got a bunch of sulfurs on our virus, right? The other thing that's really neat is that gold nanoparticles, when they're a certain size and they're spherical or rod shaped, they're colorful, right? Gold itself is, the metal is a lustrous golden color, but nanoparticles, they're red. They sort of look like wine. And that's because it's a phenomenon where gold sphere, as it's just too small, when it gets small enough, when light hits it, the electrons on the outside of that sphere start swashing back and forth with the same, and that swishing back and forth, that oscillating, actually causes it to absorb some of the light that's coming. It absorbs the blue light. And so what you end up seeing is this really wine red color. And so this is what we saw. Basically, you can start with the virus in a solution with 50 millimolar gold tetrachloroauric acid. So this is just gold acid, right? And you just mix it. And then you add a reducing agent. So you add sodium borohydride. So it's going to take that gold down to gold metal, gold 0. And it's going to specifically grow, we saw it, in the pores, because the gold's not going to go just anywhere. The gold needs to grow. And it does this because it's a crystal. And in crystals, they start attaching to gold. And the gold attaches to the sulfur. Another gold atom sees that gold there, attaches to that gold, because gold likes to form metallic bonds. And then they expand and they expand until they fill the pore. And you can see they actually fill the pore. They fill the pore. And then once they get to that maximum size, they're done. You have more gold growth that happens in solution, and that just precipitates out. All the gold that grows to that confined space. So the gold nanoparticles we're using end up being 3 nanometers. And so those in theory should be small enough to pass through the kidney, but they're now in a virus that's 30 nanometers. So those should be big enough to passively accumulate in a tumor. Okay. And we did ICP-MS. I wanted a nice picture to show that data, but ICP-MS data is kind of boring. But basically the end conclusion is that what we end up with is essentially gold with Q beta with an average of about 10 gold nanoparticles, right? And then the question is, how does a 4 or 5 nanometer gold nanoparticle fit into this 2 nanometer pore? And we did some computational modeling to sort of understand how this works. And we modeled the pore structure and the disulfides, and we set its distance based on crystallographic data. So the virus itself is structurally known because of crystallography, and we atomistically know where every atom is. And then we sort of modeled a gold nanoparticle sitting on it. And essentially what you have is this large naked sphere of gold sort of cradled into the protein. And this is really great for what we want to do, because we want to hit the gold with a laser, make it hot, and that thermal energy should get transferred directly into the protein. We don't want the thermal energy really to go into the solution. We don't want the thermal energy to actually heat anything up other than the thing that it's attached to, the actual protein. So we did the experiment again. We took the virus, we loaded it up with doxorubicin and passively accumulated. We grew gold on it, and then we tickled it with a laser, right? And then all the doxorubicin comes out. And we can see that basically because we see that Q beta plus dox is non-fluorescent, just like I said. Free dox itself, the free doxorubicin in water, is fluorescent. And we show that here. And so we just monitor the increase in fluorescence. So this is fluorescence intensity based on laser pulse, all right? And so this is an interesting result. We didn't just shoot it with a laser and just let it heat up, right? What we did was we shot it with a laser for only, this says 6 nanoseconds, but it's actually 6 picoseconds. And what that does is it heats just the surface of the gold, right? And that high temperature on the surface of the gold causes the proteins on the bottom and the water on the outside to get really, really hot. And that can cause a huge thermal expansion around the gold nanoparticle. But gold has a metal, solid metal core, and metal, as you know, is a great thermal conductor. And so all the heat on the outside quickly moves to the inside, cooling the surface, causing everything to suddenly collapse. All that gaseous water vapor collapses down the protein, condenses really quickly, and it basically destroys everything very locally. It doesn't heat the solution whatsoever, but everything around the vicinity of that gold nanoparticle is now destroyed. And we kind of see this using circular dichroism, and probably one of the worst uses of it. Essentially, we just watch the protein fall out of solution when we hit it with a laser, okay? And we did this in a cell. And so this was a fun little experiment where we looked at a cell, and we're looking at essentially the top side is a bright field image. So this is just a black and white photo of the cells. And then the bottom is through the, we call it DOX. It's the DOX channel. It's the red fluorescent channel, okay? And if you just give cells doxorubicin, so you just incubate it with, you see red fluorescence in the cell from the doxorubicin. So that drug is floating around inside the cell, and these cells now are going to die, okay? Doxorubicin is a very toxic molecule. We give cells Q beta gold nanoparticles. There's no fluorescence there at all because there's no doxorubicin in that sample. That's our control. And then we loaded a sample up with doxorubicin. And again, it's not fluorescent because, like I said, when the doxorubicin is in the virus, it's not fluorescent. And so we see nothing here. Now, what's cool is we shoot it with our laser. We shoot all these cells with a laser, right? And the blank cells, you know, this is the laser is on and off so fast, nothing gets warm. Blank cells are totally fine. They are okay. The doxorubicin cells are very sick, right? Because they're now, the doxorubicin is killing the cells. They're not doing well. The gold nanoparticle cells, they don't look terribly happy for some reason, but they're all alive. And there's no fluorescence, right? Because there shouldn't be. And the Q beta gold nanoparticles, they also look okay, even though now you can see the doxorubicin because it's just come out. And eventually those cells will die too. And the Q beta gold nanoparticle cells actually don't. So to show how close, how specific we are, we can actually look at targeting the cells. And so Candace came up with this really neat experiment where she made a pinhole and put it on top of a jar of cells, macrophages, because macrophages are really big cells. And so that makes it really easy to see. And we can draw a line here. The green line is essentially the edge of the laser beam, all right, that comes through there. And we can stain the nucleus with host. So these are the nuclei of the cells. We grew all these macrophages really tightly together. And we shot it with a laser. And you can see the laser only induced doxorubicin release within the beam path. You really see nothing outside of the beam path. And, you know, lasers do actually have, they're not like perfect points of light. They actually have a little bit of a spread. But essentially, you don't see any significant change because the laser is not heating the media. It is only releasing the drug inside of the cell. And if you do an overlay on top of it and zoom in, you can really see how the doxorubicin has been released in the cytoplasm of the cell, only in the area where that laser hits, right? And so this gives us very precise control over drug release, even for a molecule, even for something that's already inside the cell. So this is already inside the cell, and we can now use something to reach in and release the drug, okay? And now, so that was really neat. And that was sort of our first generation design. One of the problems with gold nanostructures, of course, gold nanospheres specifically, is that they absorb green light. And you're probably, I mean, you're all very smart people. You know that green light does not pass through you. That would be really weird. But you may not know is that near infrared light. So the light that's just a slightly redder than red, just slightly outside of your visual abilities. So you imagine red and then just imagine red, so red you can't see it. That actually does go through skin very well. And it can get into the tissues underneath the skin, because it's just sort of this nice little optical window where melanin doesn't really absorb that light. Hemoglobin doesn't really absorb that light very well. And, you know, so it's sort of a transparent window. And near infrared light is therefore a good way to sort of activate things a little deeper into the body than what green laser, because you shoot a green laser on skin, it's not going to go in. It's just going to hit the top of the skin, and it's going to warm the melanin in your skin. And so what our idea was to create something that could absorb near infrared light and then cook a tumor, right? The problem is, of course, just cooking one tumor doesn't really, I mean, that's fine. You might as well just do surgery if that's what you want to do. Because if you've got a tumor that's just underneath the skin and you kill it, okay, that's great. But you can just cut that out. You can do that in an inpatient setting. There's really no reason to actually just cook a tumor that you can readily access by a skin. What we were really interested in is using those sort of conveniently accessible tumors to initiate an immune response, to create an immune response that would tell the body, you got a tumor, right? You should kill you should make, T cells aware of this, and the T cells go off and then they police all their tumors. The problem is, of course, tumors are immunologically pretty dead, right? The tumor is allowed to exist because your immune system has simply decided that it was not a threat. It has incorrectly decided it was not a threat. And so a number of important components of the immune system are reporting wrong information. And it's because the cells themselves are also sort of promoting that through secretion of various enzymes and small molecules that just sort of tell a very big important cell in the body called dendritic cells and macrophages, don't worry about it. And they don't. So dendritic cells, which are responsible for going around, dendritic cells aren't really killer cells. They go around, they find things that are wrong, and then they tattle on them to T cells. And T cells are sort of like the police of your immune system. And they're not doing their job. Macrophages go around, they like to kill cells. They'll also tattle, but they're good at killing cells. And the macrophages that are in tumors, they are actually saying this place is great. No problem here. In fact, you should come and let's have a party here. It's so nice. So a big part of cancer immunotherapy is trying to tell the body, you gotta do something about this. We got to invert the immature dendritic cells, wake those up. And then we have to get them to go tell T cells. Because there's also this other class of T cells called T regulatory cells. And then they're, even if a dendritic cell comes and like, hey, I think we got a problem, the regulatory T cells are like, nah, you don't, you're good. Don't worry about it. And they take a little money, Chicago politics out there. Now, what is great about modern therapies, we now know this. We now know that an important part is if you can poke a tumor somewhere accessible and it creates an immune response, that your immune system will then go fight the cancer everywhere. So I've kind of already gone over this, and this is probably a little bit more deeper than most undergrads who haven't had an immunology course. But essentially, the idea is what we want to do is sort of fool the body into thinking that it got burned. Because when you get burned, you see pus swell, your burns, you get pus because that's an immune response. But your body's smart enough to know not to attack its entire self if you get a burn. Really, what you need to do is convince your body that you burned yourself and you got an infection. And so that's going to start the whole immune response. And the idea is, while it's there, while your immune system is there, hopefully it will see the cancer, because you finally made that area vulnerable to immune recognition by not only creating an injury, but then throwing in something that looks like a virus. And then because it looks like a virus, the immune system is going to treat it like a virus. Okay? It's not really a virus. It can't replicate. It can't do anything. It just looks like one because it's made from one. And so that was our idea, was to actually heat a tumor up so much and then have so much virus crud in it that the immune system then attacks it. And so we've combined these ideas. That was a bad animation, but you see the photoimmunotherapy. And we did not invent photoimmunotherapy. Other people have. They've used materials that are very common, that are known. Carbon nanotubes are really great at absorbing light. They don't leave the body. And they're pretty toxic. But they do work. This idea came, in fact, this was an idea I saw at a seminar, and I was like, wow, that's a great idea. I don't like the carbon nanotube, but I really like that idea. And this is how this whole project actually began, was I was like you, watching a speaker thinking, that's neat. And I think I can do it better. Which I hope you see this and you have the same idea, you can do this better. And I hope you do. And then they've done it with liposomes. Liposomes are better, in a sense that they're not as toxic. You can make them out of more biocompatible materials, but liposomes are kind of a pain. They're fragile and they're hard to get circulating. And none of this is inherently immunologically active. Like a liposome looks like the surface of your own cells. So it's not going to really trigger any sort of immune response. So we came up with this. All right, the idea is instead of putting a gold nanoparticle, we're going to put this organic dye called croconium. And a croconium dye is really interesting. You can see the chemical structure there. That's a really bizarre chemical structure, right? And it's really conjugated. And usually rigid conjugated things like this are fluorescent, but this isn't fluorescent at all. And if you remember from your PChem, right? So if you have energy that goes into a dye and it fluoresces, then the energy comes back out as a photon. But if there's no fluorescence, all that energy comes out as a squiggly line back to the ground state as heat. And so because this dye is so efficient at taking in photons, it has a really high extinction coefficient, it's also really, really good at generating heat. In fact, it's even better at generating heat than gold nanostructures, and I'll get to that in a minute. And so this is sort of our overall design. The idea down the road is we're going to make a virus that absorbs light, gets hot, cooks the cancer starts dying, and the cancer releases something called damage-associated patterns. And those are cells, those are biomarkers that tell your immune system something catastrophic has just happened, please send in support. That will bring in dendritic cells, that'll wake the dendritic cells up, they'll come in, they'll like look for stuff, maybe some antigens, maybe a bacteria, maybe a virus that they can go tell the T cells to go kill, or tell the B cells, the T cells go tell the B cells to go work out an antibody solution. And you get this idea goes down to the lymph nodes, and there, once it's in the lymph nodes, then it essentially propagates throughout the entire body. Your entire body starts to have now these T cells, these cytotoxic T cells called CD8 T cells, and they're the executioner. You know, the cancer has been found guilty in the court of the lymph node, and they sent out cytotoxic T cells to go kill the offending tumors everywhere in the body. And so, yeah, Arzu, who just graduated, she started her job at Pfizer with no postdoc. That's really great. So, you know, go from your PhD that you only completed in 4.5 years to a job at a Fortune 500 big pharma company is really nice. But she worked very hard to do this, and she made this dye using fairly simple organic chemical transformations that are nothing particularly complicated. And then she bioconjugated it to the surface, to one of those surface lysines that I told you, not the sulfurs, but the nitrogens, so on the surface of the cupeta. And essentially what that does, we had to make the activated ester. I forgot about this part of the slide. Essentially, you make the virus, and the virus is actually now greenish, right? So it's green because it absorbs really far near red light. You can see it. So these are some gels. So this is SDS gel that essentially shows slight migration because the dye is big. And these are an agarose gel. And what an agarose gel is, it's a dense network. Essentially, it's whatever kind of chemistry, they're basically like fancy TLC plates, right? And so we can see that they're all intact, and we have better systems. We just look at it with our electron microscope. We have a TEM and DLS, and we see that it's still a virus, right? We've just attached the dye to the surface. And what we found is we can heat it really hot. Like if we have a photophage, we can heat a solution with just that virus by shining a near-infrared laser on it to 60 degrees. In fact, we can heat it hotter with the virus than we can with the dye. And that's because what happens when you put the dye on the virus is it causes the ground state to spread out. So this really nice, the peak right here, this is the normal absorption of the dye. When it lays down on the virus, for some reason, we're not quite sure why, like the vibrational modes just spread out. And so now your extinction coefficient at 808 nanometers, which is where medical lasers are that you find in the doctor's office. If you go to a doctor's office and they do ICG or any of the other photoablative things in the doctor's office, 808 nanometers. Now you can see the extinction coefficient has gone up. If you remember Beer's-Lambert law, be the same concentration of dye has the same absorption, but you increase the extinction coefficient. I'm sorry, if you the same concentration, same path length, but you increase the extinction coefficient, that same amount of dye will absorb more of that light. And that makes it heat up much more effectively because all the light that goes into it turns into heat. And so you can really, really heat up the dye. You can really heat up the 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 virus. And now the cool thing is because this virus is a phage, right? I told you it was a phage. It infects bacteria. It has to survive being outside. It has to survive all kinds of things. You can heat it up to that temperature and cool it down, and it's fine. It doesn't mind getting that hot. In fact, we once, for a reviewer, asked us to just heat it up again and again for a week. And it's fine. It doesn't mind that. It can do that all day. Right. And so the size distribution doesn't change. Nothing really physically changes it, but this is the heating and then cooling cycle we can do over and over and over again on this dye, on this virus. And the virus is just fine. And the reason that's important is because we need it to look like a virus. We need it to stay a virus as long as it's in the tumor, because that is going to bring the immune system to bear, right? And so we did this relative comparison. This was a neat little study that we did to sort of show that the photothermal efficiency of the photophage is higher than that of gold nanorods. And so instead of being a sphere, you can make gold nanoparticles of rod shape. And that elongated axis actually allows them to absorb near infrared light. But they have to be pretty long. And so, but if they're the long, they have this, they're gold. They have this wonderful heat capacity. And yet somehow our little virus is more capable. It can heat the same amount of solution, in other words, faster and hotter. So it's a much more efficient photothermal system. And so this slide essentially says our photophage is not toxic unless you heat it. If you heat it, it kills like everything, which it should, right? So the constituent components, the proteins of the virus and the dyes, none of them are toxic. And so we went in vivo and we looked at a mouse model and we implanted give that away. And we implanted a mouse version of a breast cancer. 4T1 cells are essentially triple negative breast cancer. They're not immunologically hot cancers. Like there's really nothing about triple negative breast cancer that really looks like cancer to the immune system, and it's very hard to treat because of it. And so we wanted to see if we could ablate it. We could heat it up so much that the cancer just died, and then induce an immune response. And so long story short, we implanted it and we monitored tumor weight after treatment. And we found that treated tumors grew much slower. So we were able to kill a significant amount of the tumor before it could really spread. And that allowed, so our treatment group is this green dotted line. All our controls are the solid lines in these first things. And you can see this green dotted line. These are the mice. Every time it steps down, a mice dies and it gets its wings. And you can see, albeit this very aggressive cancer did kill all of our mice, but they lived significantly longer than if we just gave them Q beta, if we just gave them the dye, if we just tried to heat them with a dye with no Q beta. And what was interesting is even if we heat them, we just take the dye and we pump them full of dye, and then we heat them to about the same temperature as the Q beta crock, it still didn't work very well. And there was something about the Q beta itself that was promoting this. And we suspected, we think it's promoting a more anti-tumor effect. And 4T1 cells, just like in human cancer, in a mouse, they metastasize, and they like to metastasize to the lungs. And so Arazu pulled the lungs of the mice out of our control groups, and you can see the white dots are the tumors on the PBS control, the purely croconium treated. So this is just the dye. There's no Q beta involved. It's just the dye. And if we put enough dye in there, we overcome, we just utilize the other part of Beer-Lambert. We just pump up the concentration a lot, even though it's got a lower extinction coefficient, we can still get the same absorbance. And there's still a lot of metastasis. And then if the photophage, you see some spots where there's definitely some metastasis, but it's dramatically lower. It's dramatically lessened the metastatic burden on the mice. And we wanted to see if this was because of an immune response. And so Arazu essentially looked at 3 components. One, how does the virus impact the percentage of mature dendritic cells. So dendritic cells that are matured because they've encountered an antigen. What we find is that the virus itself does promote dendritic cell formation. Now, the virus by itself doesn't actually treat the tumor. All the mice die very quickly with just the virus itself. It has no therapeutic effect. But you see it has the same action even when you heat it up. You heat it up, it cools down, dendritic cells are still activated. But if you just heat up the tumor, that doesn't activate dendritic cells, because there's no reason for your body to get really bent out of shape over something that got burned. Dendritic cells are there to sort of initiate a systemic immune response. And in a burn, you really need just a local response. You just need to make sure there's no bugs in there. Now, the T helper cells, these are the police. So these are the ones that are going to come out, and they're ones actively monitoring to see if there is going to be a dangerous infection or cancer. And we get a significant increase in the percentage of those only in the combination of virus and heat, which means you need both in order for these T helper cells, these CD4 cells, to come out and sort of adjudicate whether or not there is a problem. And then finally, we looked at T regulatory cells. And these are a special kind of T cells that just tell every other cell, then just don't worry about it. The immune system's fine. And these cells are really upregulated when you burn yourself, because there's absolutely, again, no reason to launch a systemic immune response that'll make you extremely sick because you burned yourself on the stove. That doesn't make sense, which is why just cooking a tumor actually sometimes is worse for cancer, because it actually shuts down the immune system. But when you have it in a way where you not only burn it, but you also make your immune system thinks it's infected, we find that the T regulatory cells are actually quite low in the tumor. They are not elevated as a result of the thermal treatment. So what we're doing next is essentially moving on to synthesis and conjugation, and we're looking at the stability. And we've already assessed in vitro and cytotoxicity. It's very low toxicity. And now, of course, the next step is to simply look at improving the immunogenicity of these things to increase the immune response by adding something that actually, so the virus itself just sort of fools the immune system into being more vigilant. Because a virus is empty, we can put anything we want in it. We can put molecules that once the immune system becomes vigilant, we can put molecules in them that actually make the immune system very angry. And, under normal circumstances, you don't want the immune system to be angry. But when it's inside of a cancer, you need the immune system to wake up by hook or crook. You need that to start working. And so that's our next step, is to sort of turn up the volume on the immune response. And that's it. So this is the people that do the work. I mostly sit in my office giving talks like this. This is, I said, Arazu. So she just started her job at Pfizer right after she got her PhD. Olivia was a major help in this project. She started her job at Merck very recently as well. And so when people that graduate out of historically done very well for themselves. I have a broader slide of more students than where they are right now, but these are the students that helped on these projects. And so I'm obviously very, very thankful for their contributions. And Candace, who started the project, she's now at VacSite. She's a conjugation scientist now making vaccines. And so I am happy to answer any questions you have.
All right, so what are our questions, everybody? Well, I have a question. I'll start the questions rolling here. Are there any examples of using viruses like you do for drug delivery that, in humans, like, in clinical trials or anything like that?
So there are FDA approved viruses for gene delivery. And so those are used pretty extensively. Oncolytic viruses. So they have viruses for AAV, and this is the adeno-associated virus. And then they have a number of, what's the other one? Lentiviral vectors. And so that one's in the, I can't remember what it is, used in specific cancer, but they are used for gene delivery. The Johnson & Johnson vaccine is, of course, a viral vector used to deliver DNA. And so that helps produce the spike protein. So in some sense, using viruses for just drug delivery is probably a bit of an overkill for the technology, but they've gotten a lot further in gene delivery. Virus-like particles outside of that, of course, have been used for a while as platforms for vaccines. So they've been used to, so the sort of scaffold to attach antigens for things like HPV as a vaccine candidate, for instance. So the immunological response that you get from these has been pretty well studied, but you can also do delivery of genes. So yeah, there are actually now quite a few viruses that are clinically approved or have approvability to treat a number of disease or act as vaccines.
Okay. All right. Does anybody else have any questions? Well, it looks like we're a pretty rowdy crowd here, so I guess that we'll thank Dr. Gassensmith for his talk today. If you want to contact him, the flyer that I sent out has his QR code. Will that go to your website? It goes to your website? Okay. So feel free to reach out to him if you're interested in any of this stuff. It was really a really interesting talk. Thank you so much for coming and talking with us.
Let me put my website in the chat there. That's not the right one. There you go. That's the right one. Gassensmith Lab. There you go. Yeah. And if you want to talk to Ryan, she's now outside. So yeah.
Well, I'll stay on for a while. And hopefully she can pop in.
Yeah.
All right. Thank you, everybody, for coming to our Weisenthal colloquium. Bye-bye.